Hello and welcome to our presentation on the cultural and ethical considerations of human embryonic stem cell research. My name is Flynn Donnellan. Other presenters today are Matthew Lee, Lindsay Larson, and Jaden Chavez. So to get started, a little bit of background. There are three major categories of stem cells. We have the totipotent stem cells, pluripotent, and multipotent stem cells. So here's a graphic just showing kind of that pattern of differentiation that you see among the stem cells. So you see start with totipotent, move to pluripotent, move to multipotent, and then of course the cells of our body. So totipotent, they are the most versatile types of stem cells. They can make any of the 220 types of cells found in an embryo, as well as the extraembryonic cells. And after roughly 4 days, the cells begin to differentiate into pluripotent stem cells, and this is from fertilization. So you imagine there's only about 4 cells, and that short window is that time frame that you have totipotent stem cells. So as we get moving here, we're now getting into the pluripotent, so a little bit more differentiation. These cells are derived from 5 to 7 day old blastocyst, so about 100 to 200 cells, which involves the destruction of the human embryo. And these cells can be taken from aborted fetal tissues. They can also be grown in vitro or in a lab and targeted to differentiate into specialized tissues, and they form any of the 3 germ layers in the human embryo. While not as versatile as the totipotent stem cells, pluripotent stem cells are still able to differentiate into nearly any human tissue. Next, we'll get into the multipotent stem cells here. These cells are more limited in what they can become. So, for example, if you have a hematopoietic stem cell, that is a multipotent stem cell that differentiates into several different types of blood cells, meaning this stem cell can be taken from the blood and can differentiate into specialized cells of other tissues. These cells are hard to isolate, and they cannot be expanded in vitro, making it hard to culture and create large quantities outside of the patient. So that kind of builds that basis of our ethical dilemma here. Here's another image just to kind of show you the progression from totipotent to multipotent stem cells, and also give you a little bit more information, the pros, the cons. You can see we have like the ethical issues with the totipotent and pluripotent stem cells, whereas the multipotent, are mostly derived from, a born, human being. And finally, here is a video just to kind of wrap everything up and give you a little bit more of a clear picture on what stem cells are.
Imagine 2 people are listening to music. What are the odds that they are listening to the exact same playlist? Probably pretty low. After all, everyone has very different tastes in music. Now, what are the odds that your body will need the exact same medical care and treatment as another person's body? Even lower. As we go through our lives, each of us will have very different needs for our own health care. Scientists and doctors are constantly researching ways to make medicine more personalized. One way they are doing this is by researching stem cells. Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine, in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease. Using stem cells to replace damaged bodily tissue is called regenerative medicine. For example, scientists currently use stem cells to treat patients with blood diseases such as leukemia. Leukemia is a form of cancer that affects your bone marrow. Bone marrow is the spongy tissue inside your bones where your blood cells are created. In leukemia, some of the cells inside your bone marrow grow uncontrollably, crowding out the healthy stem cells that form your blood cells. Some leukemia patients can receive a stem cell transplant. These new stem cells will create the blood cells needed by the patient's body. There are actually multiple kinds of stem cells that scientists can use for medical treatments and research. Adult stem cells, or tissue-specific stem cells, are found in small numbers in most of your body's tissues. Tissue-specific stem cells replace the existing cells in your organs as they wear out and die. Embryonic stem cells are created from leftover embryos that are willingly donated by patients from fertility clinics. Unlike tissue-specific stem cells, embryonic stem cells are pluripotent. This means that they can be grown into any kind of tissue in the body. A third kind of stem cells is called induced pluripotent stem cells. These are regular skin, fat, liver, or other cells that scientists have changed to behave like embryonic stem cells. Like embryonic stem cells, they too can become any kind of cell in the body. While scientists and doctors hope to use all of these kinds of stem cells to create new tissue to heal your body, they can also use stem cells to help understand how the body works. Scientists can watch stem cells develop into tissue to understand the mechanisms that the body uses to create new tissue in a controlled and regulated way. Scientists hope that with more research, they can not only develop specialized medicine that is specific to your body, but also better understand how your body functions, both when it's healthy and when it's not.
When looking at research, our pluripotent stem cells are going to be our most valuable. Totipotent stem cells, they can make any of the 220 different cell types found in an embryo, but they have a very short life phase and they almost immediately go to the pluripotent cells, which is exactly why we are most concerned with those in research. The benefits of these stem cells are that they have the ability to undergo self-renewal to generate more stem cells. They can also be grown in large quantities in a lab environment. They are taken most of the time from aborted fetus tissues, which is kind of going to be where our against argument comes into this. One of the biggest benefits, though, with these stem cells is that they have the ability to differentiate into any of the specialized tissues in our body, such as muscle, neural, or epithelial, which can be extremely helpful when we're testing new drugs for the safety and effectiveness. Instead of using an actual person for clinical trials, this way we're not risking a human life. This, in turn, helps us to increase our understanding of disease processes and the best ways that we can intervene from a medical standpoint. It gives us the ability to observe neurotissue forming and see how these disease processes develop, as well as what points are more beneficial to introduce cares and medications. These tissues can also replace human tissues in the event of a life-threatening disease through what we call regenerative medicine. With regenerative medicine, we're using these artificially grown tissues and organs to replace tissues in diseased patients who suffer from things like type 1 diabetes, Parkinson's, ALS, Alzheimer's, heart disease, stroke, burns, cancer, osteoarthritis, and many more. Ideally, these replacements would be personalized enough to prevent rejection in our patients. It goes without saying that this type of research can make a huge impact on medicine and healthcare as a whole. However, we're not sure yet on the exact dollar amount. For patients who suffer from things like diabetes, imagine the amount of money that they can save on treatment of insulin and all the costs associated with things like glucose strips, pumps, and visiting their endocrinologist. They also won't have to worry about mismanaging their disease or constantly having to monitor their glucose, which gives them more freedom to enjoy just living their life.
And that brings us to the central tension of our ethical dilemma. Arguments for and against the use of pluripotent embryonic stem cells are not tied to the usefulness of the treatments being considered. Instead, it largely revolves around the ethical considerations of when life begins and when do we award rights as a human person to the unborn embryo. Vladislav Volarevic and colleagues phrased the question succinctly in their 2018 article addressing the topic. Whether or not it is morally acceptable to pursue novel therapies for curing illnesses, if it comes at the expense of destroying an early human embryo. Embryos develop personhood in a moral and ethical sense at a later stage of development, according to the pro-argument. But there are nuances to the pro-arguments. Most people on the side supporting stem cell research don't think the embryo and blastocyst are just any clump of old cells. Rather, they still believe that an embryo still deserves respect as a potential human person, even though it can and should be used in good conscience for research, as long as there is scientific justification for its use, careful oversight, and informed consent from the woman or couple donating the embryo. If we assume that this idea is correct, that personhood is bestowed on a fetus later in development, then there should be no ethical or theological violations towards destroying an embryo and using it for research.
And with that, we move into the against argument. The against argument to stem cell research, specifically human embryonic pluripotent stem cells that we've been talking about. Embryos become human persons in a moral and ethical sense at fertilization, at when the sperm and the egg meet and form a cohesive DNA structure. So at that point, it becomes a human person in the against argument, and thus should be granted all the rights afforded a born child. In this argument, any action that we take to terminate the embryo is morally equivalent to terminating a living, breathing, born child. And so in this argument, ethical principles that would be violated, if this is correct, would be non-maleficence and beneficence. And we'll explain that here in the next slide. So in medical ethics, non-maleficence states that the procedure must not be for the harm of the patient, and beneficence means that the procedure must be for the good of the patient. Now, in the against argument, we're assuming that the embryo is itself a patient, and thus both of these ethical medical principles of non-maleficence and beneficence are then violated by then killing it. Bringing in theology and sort of the religious position that tends to go along with this, we see that there are some inalienable rights to life that are granted by a creator to human persons. So this is a common theological principle that the against argument tends to be associated with. So if the human embryo is in fact a person at fertilization, then termination of that embryo for stem cell research will then violate the theological position of a right to life. Not only religious implications, but here are some cultural, political implications that kind of get wrapped up in this dilemma as well. Here you can see that there are a couple different political cartoons throughout the past couple of years that sort of accentuate the tension that tends to go along with political choices as well. You can feel free to pause and check these out if you'd like. Slightly humorous cartoons. And moving on, our possible nursing implications, how we would be involved in stem cell research and clinical treatments moving forward. We kind of looked at the California Institute of Regenerative Medicine. They proposed a concept of a hybrid nurse that would have extra training and certification that would allow them to administer stem cell therapies, as well as monitor the patients for the intended drug effect, as well as the adverse drug effect. And that's how nurses could possibly be involved in this ethical and cultural dilemma in the future. Thank you very much for being with us through this presentation. Here are our resources if you find yourself with plenty of free time and would like to read them. If not, thank you very much.